This method can help answer the spatiotemporal regulation of a gene of interest during dynamic biological events such as immunity in intact Arabidopsis leaves. The main advantage of this technique is that it allows us to capture the spatiotemporal dynamics of the promoter activity in soil-grown intact plant leaves over a few days.
To begin this procedure, fill a plastic cell plug tray with autoclaved soil. Sow 1 transgenic Arabidopsis seed into each cell. Transfer the tray into a growth room that is maintained at 23 degrees Celsius and grow the plants under continuous white light conditions for 2 to 3 weeks. 2 days prior to pathogen inoculation, streak the pathogen-carrying AVR-RPT2 from a glycerol stalk onto NYG medium that contains rifampicin at 100 mg per liter and kanamycin at 50 mg per liter. Incubate at 28 degrees Celsius for 48 hours. Using plastic tips, harvest the bacterial cells that appear on the surface of the medium. Transfer the bacteria to a plastic tube containing 10 millimolar magnesium chloride and resuspend them. Then measure the optical density of the solution at 600 nanometers. Adjust the final concentration of bacterial cells to 100 million colony-forming units per milliliter, which normally corresponds to an OD600 of 0.2. First, carefully cut out a cell plug containing a 2 to 3 week old plant, making sure not to damage the plant.